Okay, so we'll keep moving. We've one more session before the break. So I'd like to invite Federico to the stage as the moderator. Federico is Associate Professor at the Instituto Superior Técnico, also a PI at the Institute of Bioengineering and Bioscience in Lisbon. He's also the coordinator of FEAS, and he's also an H4P. So I'd like you to introduce the speakers, please, Federico.
Okay, thank you very much. It's an extreme pleasure for me to be here. I define myself as an innovator and educator, and the session about today is all about technology. So we will see how can we use the technology innovation to overcome adoption barriers, and we have the honor to have three speakers. The very first speaker, I'll introduce one by one. The very first speaker is Professor Mark Fenelon. He's the head of the Food and Research at Teagasc, Food and Research Center at Moons Park, and is responsible for, has been responsible for the program of 2 research centers. Previously, he has been working with the infant pharma industry, and now is working in different processing systems, both in dairy and plant ingredients. His presentation today is titled Simplify Fractioning Process and Byproduct Utilization by Protein Crops, and he will present a work that has been done through the Valpro Paths, and that has been very important because he refined and simplified the fraction techniques of high protein crops. In this way, you can generate very high value ingredients. So it's all about what we are here doing, that is making simple solutions for complex problems. I ask Mark to join the stage. Thank you very much.
Thank you very much, and I'm delighted to be here, and looking forward to meeting colleagues from GIANT LEAPS. I suppose the first thing is, I know everybody's going to say to me that I didn't simplify it, I made it more complex, so apologies for that. So I suppose that some very interesting questions have come up, and I worked in the infant formula sector and I worked in the dairy sector as well for years. Don't worry you, and I won't mention too much about dairy, but I think it's important to note that if you think that milk contains 3.6% protein and 0.8% of that is whey, 0.8, and we fractionate that into now ingredients that are getting 25,000 a tonne because of a whole lot of different reasons, including the GLP-1 and so on and so on. You know, the question comes, why wouldn't you do that with fava bean, which is 26%? So Fenelon, you know, it makes a very obvious question, so that's what intrigued me. The other thing is that milk doesn't contain fibre, and that's now an ingredient that's also growing. So the conclusion of my talk before I start is that I'm talking about ingredients here, and ingredients will build a nutritional base then for formulations into all sorts of different products. So what I'm going to show you here, and I'll go quite fast because at the time, are some of the products that we made and some of the processes also that we use. But remember, we're talking about fractionation into ingredients. So unless you can get not just the protein, but if you think what they do in the dairy industry, we have the cheese, we have the whey, we have lactose, delactose, permeate, and go on and go on and go on. Unless we can do that on the plants, the business case is more difficult to stack up now. So André, you know this better than me, but that's what we're trying to do. So that's what we're talking about when we're talking about fractionation into ingredients. This is not a real newspaper, please don't look for it. I made this up on the computer. But just what I'm going to focus in a little bit is on dry and wet fractionation. Talk a little bit about high shear processing, might touch on hydrodynamic cavitation, and just some of the products that were made. I'm going a little bit fast because of the time, so come up to me at the break if there's any specific questions. But first of all, I want to make the point, and Ewan made this earlier very well, and this is what made Valpro so interesting for somebody like myself, because the variation in composition, it's about that. It's the same in any product. It's not just the yield and so on, but what is the protein content, what is the starch content, and what is the fibre content. And even if you look between Denmark and Ireland, Denmark have a higher protein content than we do, but the variation in varieties within that. So if Vladimir is trying to increase his logistics on a high protein isolate, he wants the variety that gives him more protein here, and he also wants the one that yields higher. So that's an important point. So the composition is really important when you're talking about fractionation. There's two types of fractionation process that you hear. One is a wet process and the other is a dry fractionation process. They're very different and they give you very different ingredients. For instance, the wet process, which I'm going to talk about a lot here, is a full fractionation into starches, fibres and so on, but it gives you an isolate ingredient. So you can get your high protein ingredients. Whereas the dry fractionation, there's no water involved. The powder is milled very fine powder and separated based on density using cyclones. So you get a 60% protein ingredient, and then you get an enriched starch ingredient as well. So it's important to know that, because if you're trying to formulate something with a dry fractionated one, you have starch, fibre and other innate nutrients in there that's not in the isolate. So some products then you won't be able to use that for. Even though the ingredients have a different price range and so on, there is a big difference between what the two fractionation processes. So just maybe at the outset, one very nice output that came from Valpro, we designed a lot of the processes and parameters for processing to give us the functionality within the ingredients for different foods and beverages in Valpro, but it allowed us to leverage extra funding then to build a full commercial scale, pre-commercial scale pilot facility in Ireland from this. So we're using the technology then and what we learned in Valpro and so on, and we got the money then leveraged from Enterprise Ireland to be able to build a full scale. So we now have that capability. So this gives us fibre, starches and so on as well. But again, and this was one of the recent workshops Tamara I spoke about, and these products then were made through Valpro. So there's a whole range of different products here. Some of them were made with the protein isolates, and some of them were made with the starch fraction or the starch fibre fraction. And we used an innovative technology which I'm going to show later on how to dry that starch fraction, because drying the fava bean protein is one thing, but then drying the starch fraction is a little bit more difficult because of its viscosity. So I'll show you that technology. And this was a very nice fava bean cream cheese which was made in University College Cork by my colleague Emanuele Zannini, who's in the audience, and Elka Arndt, who also made a lot of different breads and different products as well. So we gave them the ingredient that we made through Valpro, and they made some very nice products. And these were very successful products, the whole range of different products that they made. This is really what I wanted to talk to you about. So this is the isolate, which is greater than 85% protein. This was made using a wet process, and this then, the residual biomass is your starch fibre fraction that's left after that. But you can go further then and separate that into starch and fibre again into new ingredients. So these ingredients here are what we use then for producing some of the products that I'm going to show you. We also looked at lupin, chickpea, and lentils and so on as well through Valpro, which I'm not covering here, but can cover on the breaks. So this is your fava bean, your dehulled. This is your flour, and then this is your starch, and then this is your isolate. And it's interesting because to use this, then you can use it by modifying the process. You can then make a lot of different functionalities within your ingredients to give you different products. This was a bar that was made by my colleague Mohsen, who's also here today. So we made 2 bars. These bars are the actual bars that he made, and these ones here as well. And what we did then, we used the yacon syrup, which was supplied by Biotanix, who's also in the audience here today, which is a very interesting high frost inulin ingredient, which was used as the binder in the bar. So we used the protein isolate from Valpro, we used the yacon syrup then as well to modify the glycemic index of that bar, which was very interesting. And it did the functionality, its rheological properties were very good. My colleague from India has told me these are very, very popular snacks in India. Again, these are made by Mohsen. So this is where we use the starch fibre ingredient. So now we use the byproduct of the protein. So again, this is back to Vonimir's point. We have the protein isolate application, but this is where we use the starch then. And these were really, really nice. I've eaten them myself. And the functionality of that then, again, through our processing, we can modify that and even change the starch fibre ratios for applications in any starch-based products. But this is kake, if I pronounced that right. Is that right, kake? But it's a really popular snack in India, and they're really nice. But we also made fava bites, fava cubes and so on, and Mohsen, he's at the back of the room down there, he can talk to you about the manufacturing of these. But again, it was about getting the functionality right, and a lot of projects fail. We talked about getting products from pilot plant up. Most of these projects fail because people won't buy them if they're falling out in a plant, if they're if they're not hydrating properly, or they're causing problems when they're processing plant. So that functionality is really important. This is a nutritional beverage. Then we made this with PepsiCo. This work was done. So it was a protein beverage, nutritional beverage. We made a standard one, and we also made an emulsified one. So the emulsification properties of the protein isolates that we used and the dry fractionated ones were very good for fava and also for pea and also for chickpea as well. So all these were made through Valpro, and the functionality was really, really good. So it matched, in many cases, a dairy protein. One thing that we did then, which is a bit of additional work we did, was we used the dry fractionated fraction then to bring down, I suppose, the cost of ingredients in it, but we did find we got some gelation after that. So we did some work on that, which I'll show you now later on. And I also want to mention the paper then Ewan mentioned earlier from my colleagues in University of Athens. So this is a really nice paper which looks at the carbon footprint of these beverages. I'm going to move on quite fast because I'm coming up to time. But this looks at the functionality. This is where we looked at the functionality. We have our starch here, and we made a whole load of products then based on modifying the processes to modify the functionality of that starch, but also then our protein here. So we can use starch pasting cells and a lot of different technologies to study the functionality of these ingredients. And these are the beverages. These were some muffins we made from it, again, successfully from the starch fraction. These are some meat crisps which were made from our colleagues from China. A really nice addition. Molson also made these, and I think the interesting thing is here when they were added into the hot oil, we were able to get the functionality really good. So again, this is made from fava bean starch fibre fraction, which is the byproduct of the protein isolate. And that's the crisps there. Here we looked at hydrodynamic cavitation. So again, we did some pre-processing to increase the amount of protein we could eventually get from our serum phase. And this technology here, the one on the right doesn't look too appetizing, gave us an increase in protein that we get from the serum phase up to 37%. So by using hydrodynamic cavitation, which applies a very high shear, we were able to increase our protein yield within the process. This technology then, which is a drying system which we bought from VOM in Italy, it allowed us to roast the powders, but it also this is what we're using for drying our starch and our fibre. So we've got a very nice, that fibre starch ingredient that we talked about, that was dried on this system. But you can also roast the powders, and we're doing some work with Kerry, who's also one of the partners in the project, and I should say PepsiCo are too, and they're here today. So we're doing this work here to modify the flavour profile. And if you look at it, you can see the roasting here, and if you look at it on the GCM SMS, then the volatile flavour profile is very, very different, and this was done by my colleague Kieran Kilcawley in Moore Park. But you can see very cleanly then there's a very big difference in the roasted powders, and you can see the colours different straight away. So again, that technology has a dual purpose. It also brings down the microbial load, which now gets you into the high-end nutritional beverage sector, because you can bring the microbial load really down by using that technology on your finished powder or on your starch fibre fraction, which is coming from your process. This fraction here, this is our wet processing, and we talked a little bit about the closer to farm gate. So this technology can be used closer to farm gate. So basically it's a processing tool, or a high shear processing tool, which you put your fava beans directly into it, and you can fractionate then, or turn them into a paste, which then can be further dried or further fractionated. And this can be done at the farm level. I'll finish up then just to say we also did some work on peanuts with our colleagues from Botanics across the work packages in Valpro. And again, we looked at different varieties here, and we were able to produce these cakes and oils, which we also made some foods with as well. So again, I'll just very quickly go to the summary here. So I suppose our summary is that we were successful within Valpro then of producing the different isolates, the different fractions, the fibre fraction, the starch fraction, but also then we were able to produce the products from it. And probably what I haven't shown here, if people are interested, is the science behind the functionality, the solubility, the hydration, the characteristics of those ingredients within the food. So I didn't have time to show it here today, but that was probably one of our major outputs of it. But it does show that it's a wide spectrum, and I think the question was very relevant about the markets for this, but it can show that these ingredients can be used across a wide range of different applications. Also the nutrient tracking tool, which I didn't cover today, which is done by Richard and myself and others, is allows us to go from the farm all the way through. So I might leave it there in the interest of time.
I know that you have lots of questions, but we hold the questions for the end and we do the three presentations together. So it's now with the pleasure that I introduce Emanuele Zannini. It's Associate Professor of Food Science of the Department of Environmental Biology at the University of Rome, La Sapienza, and is also a research professor at the University of College of Cork. He investigates and validates innovative and sustainable food systems to deliver affordable, fair, safe and nutritious food, and is also a coordinator of the Horizon Project Smart Protein Projects, and he has a research academic career full. Today, we will speak about how to cover the advances required to scale up the processing of alternative proteins, and I'm really looking forward to hear to you.
So, thank you, Federico, for the nice introduction. I will be very sharp because I know that the time matter, so leaving the question later. So, when we start thinking about Giant Leaps proposal, we were thinking, so we were a clear idea that the bottleneck was not longer the protein production, but basically how we can actually use the different protein we are knowing and how we can functionalize in a way that basically will turn in something that basically is a pleasure to eat. Because that is actually very, very important. So what we also, and then in implementing this proposal, we were also thinking that the major hurdle was actually the texture and the flavor. So in one of the work package, we're basically focusing on how to functionalize those ingredients and turning in a products that you will love to eat. So, and then in this case, basically with the Giant Leaps, we were developing scalable processing, and we were not actually using a fencing technology, because we need to make sure that basically this technology that we implement can be easily transferable to industry. And so we were using knowledge and processing that basically are well known and can be easily scalable. And then, of course, we were taking consideration not just the texture, but also the flavor, the nutritional profile, in order to make sure that basically what we are planning to develop is something that basically can really match our animal-based products. So, and basically we are focusing on different plant protein, particularly faba bean pea, lentil, chickpea, and rapeseed oat, and also on chlorella, some microalgae, chlorella vulgaris in particular, also from the honey varieties. Also, we were working a little bit on a single-cell protein using solar food ingredients, and also we were using different technology like enzymatic approach, and also biomass fermentation using food-grade fungi. And then we plan to develop different type of food product categories, meat analogs, dairy alternatives, and also bakery products, in particular developing egg replacer for cakes. So I'm gonna go through very quick to the different approach and results we got in this area, starting from the meat analogs. So as you see, we were targeting different ingredients, chickpea, faba bean. Also the key, what we saw is to integrating different protein sources is actually helping us in developing the structure and the rheology we are looking for. And besides that, also improving the nutritional profile and quality of the products. Okay. And then we see basically how also enzyme, in this case we were using transglutaminase, now how to build the network, the structure, so developing a firm structure that basically could mimic the patty burger texture. And also, of course, one of the big challenges here is to the flavor profile. So also we were using different spices like pepper, garlic, and we were able to round a little bit the flavor and to develop something that basically is really enjoyable to eat. So in this case, we prove, and also on the next slides, how extrusion can actually play a key role in functionalized protein, in particular in developing the fiber structure that basically are really looking for. And here also you see the different protein source has been used, in particular chickpea, and we assess what is the most desirable processing parameters, assessing the temperature, the water uptake, and also the screw speed. And then we figure out basically which are the best combination that basically allow us to develop this specific and desirable project. Of course, one key hard challenge here is to we really need to match ingredient composition and process, because changing the ingredient composition also will turn in a different approach, in a different functionality. Sorry, I have to speak too loud. Okay, that's what my students said. But then I told them, you know, I'm not speak too loud, so you are forced to shut up. So anyway, but also, you know, my colleagues André will talk about a little bit later. As I mentioned before, we were basically not only focusing on structuring, on functionalized ingredients in turning in nice products, but also to assess, you know, the nutritional quality of those products. So that's why also we were using N15 label faba bean protein, and we turned this into products and has been used for human intervention study. But this will be treated a little bit later. We said besides, you know, extrusion and enzyme technology, we were also using biomass fermentation, and in particular here using a different food-grade fungi. And these slides you see we were using both protein isolates and concentrate, but also flours. And it's also a very interesting approach, because we think that the less process we have in certain case, the better it is, because we can keep all the nutrients, all the co-passengers, minerals, fibers, fat. These also are relevant to make sure that basically we are delivering a balanced product. And definitely you see here, by choosing different fungi, we can actually improve, modulate the functionality, the technical functionality of those proteins. Particularly when we were fermenting quinoa flour, we were able to improve the gelatin, the water absorption, and when we also we were using cricket powder and fermented with the Plateus osteratus, Ericozoc with Oligosporus, we see here how we can able to change significantly the functionality. So, and this also means much more, let's say, emulsion capability, means I can actually use, you know, in a liquid system. If I'm able to develop a structure, so I can actually use in a solid-based system. So you see, and you see the QR code actually allow you to actually now go in details on the publication that's being produced when this topic has been presented. Of course, besides the fungal fermentation, we were also targeting lactic acid bacterial fermentation. In this case, we were actually using, by Hirta, the lentil protein isolate concentrated plus honey chlorella and based on oat drink. And then we were able to produce a very nice cheese spread analogs. Of course, with very high, quite good in protein, very low in fat, also a natural source of vitamin B12 thanks to the honey chlorella, and also high fiber content. And this is very important, because if you look on the shelf and you check on the back on the package, most of the plant-based alternative dairy are based on starch. So totally unbalanced from nutritional perspective. So this is also very important to make sure that basically we are delivering something that is delicious but also equally healthy. So again, with biomass fermentation using our food-grade fungi, we were also IRTA was also able to develop, a meat mold analogs. And the good things about it, the more this ingredient substitution was performed, and the better flavor we were able to get. So developing a mushroom flavor, a sweet taste, and basically significant reduction of beanie and wet flour of flavor. Of course, here we were also using full-fat chickpea. So the idea, we had some challenge in fat oxidation. So we were also turning in the fatting in low-fat chickpea flour. So to compensate such challenge. But definitely here you see also how fermentation such whole technology can actually stay able to manage and give a great support, a cost-effective support in delivering something that basically is still delicious. You know, I'm keeping saying delicious pleasure because as Italian, I mean, for me, food is very important. And I'm very picky when I'm assessing these products. So when I'm saying it was very good, means really, really good. And of course, we were also focusing on the area alternatives. We were assessing the feasibility of different plant protein isolates in plant-based milk production. And you see, basically, our main idea was to match the nutritional quality of milk of cow's milk, because most of the time, when you also check the nutritional score label of plant-based milk, now it's just water and sugar. So, and definitely here we were able to remake some of the nutritional composition of the different products. But then here you see also the rapeseed, even though it has a very good nutritional value, doesn't really fit for, you know, due to the low protein solubility. So definitely, as you see here, each protein has their potential functionality. I have to run because I'm talking too much. Also, another area where we were working in functionalizing protein for egg replacer for cakes. And in case here, we were actually very successful in using fababin. We were using two different enzymes, transagglutinase and alkylase, and we were able to replace the functionality that egg powder actually provides in a complex food system like in a cake, so much more increase the volume and also in particular the technological shelf life. And of course, when we were using fababin, the beanie flavor was still there, so the idea was actually using the flavor in fababin concentrate, and in this case we see basically that the bell pepper and the mushroom flavor was actually significantly reduced. So it was not another strategy to actually help in delivering something that basically is very unique. Also, this is the last slides, we were actually working a little bit on a solar food protein, in particular assessing their functionality, and we see basically that they are really good in gelling properties. So they can potentially develop a very good yogurt-like products because they are very good source for LAB, for fermentation. Actually, they ferment faster than milk. So definitely there is some huge potential in this area. So just to conclude and see basically which are the main outlook of this 4 years project regarding the food development, alternative protein are no longer a concept, okay? But a concrete pathway for a proper dietary transition. Plant, microbial, and hybrid protein are actually the successful key to mimic the different food categories. Of course, the technology, when used properly and when are combining together, are able to deliver products that basically are superior from technological, nutritional, and sensory perspective. But this can be done only when integrated science, technology, and consumers-driven innovation are put together, like in the GIANT LEAPS. Thank you very much.
Thank you very much. Again, we'll reserve the questions for together in the end. Now it's a pleasure that I'll introduce Emanuele Zannini, André Brodkorb. He's also from the Teagasc Food Research Center and from the University College of Cork, but he's more focused on the structure and functional relationship between food and food ingredients, in particularly in the proteins from sustainable alternative or plant sources. He has been the PI or the work package leader of several national large projects or European projects, such as the U-protein and in GIANT LEAPS. But today, his presentation, we saw two presentations that include products that include about food formulations, but we would like now to know what is the impact of that food processing on the digestibility of these alternative proteins and what are the consequences in consumer health. And so we are now completing our cycle of three presentations. Thank you very much.
Very good. Thank you for the nice presentation, for the nice introduction. I'm fully aware that I am the one holding you back from the coffee break. So I keep it as short as I can. So myself and Kieran Treat, we had the honor to lead the largest work package in Giant Leaps on digestion of food, and I will tell you a little bit about what we have been doing. And if I could summarize the entire work package in one sentence, that would be that the ingredients, the protein ingredients, the alternative protein ingredients and the foods, they actually performed quite well on a nutritional scale. So we walked into this project slightly worried about, that alternative proteins are incomplete and all these kind of worries. In general, they performed quite well. And the processing, so most of these ingredients, actually all of these ingredients were processed at some stage. The processing actually had a positive effect on it, generally positive effect. So what we did in the project is basically develop methods, a large amount of scientific data, and then of course outreach, and I will go through that bit by bit. So we had a relatively large team from 4 continents and 8 countries from Teagasc. Teagasc, by the way, is an Irish word meaning teaching. So Teagasc Moorpark. The second team was University of Naples in Italy, and the last team was INRAE Agropytech in France. So the first thing I want to show you is some in vitro digestion results of, this is a short list by the way, so it's 20 different ingredients, mainly plant-based ingredients, but also single cell proteins and insects. So what we did is we performed an in vitro digestion as an InfoGest method, our own InfoGest method that was recently published in Nature Protocols. And in the middle you see basically a graph of the digestibility, the in vitro digestibility of all these different ingredients, and you can see in general they're quite high. In general they're well above 70, most of them are 80 with 1 or 2 exceptions. When you drill a little bit deeper, that's the individual amino acids here. There is individual essential amino acids of the different ingredients. So that is basically compared to the need of the essential amino acids, and that's set out by the FAO. So green basically means excellent source of the amino acid, so more than 100%. White means between 75 and 100. So most of them are green or white, with some exceptions. There are some red, but most of the ingredients here, these for instance, or the isolates, they have very good digestibility in terms of amino acids. And here on the right-hand side is the Dias score. Dias is basically a digestible, indispensable amino acid score. So we went one step further. We have very sophisticated in vitro digestion models, and I'm not going into details. It's just to understand what is actually happening during the digestion, gastric digestion and intestinal digestion. We look at the step-by-step digestion, solubility, and what remains at the end of the small intestine. We also tested some of the foods, so not just the ingredients, but the foods that Emanuele introduced. So the meatballs, meat analogs, protein beverages, and the fermented yogurts. And this is just an example, of a recent publications. We basically look at the meat analogs and see how they perform cooked or uncooked during in vitro digestion. And that's the same table as in the last slide. So the essential amino acids of the different blends of the meat analogs, and they were blended in light of optimizing the delivery of essential amino acids. And here you can actually see, some of them are performed, super, really, really good. Okay. The second aspect, and that was looked at by my colleagues in the University of Naples, was the anti-nutritional factors. So anti-nutritional factors are compounds that interact with the digestion process, okay? And what they did is basically they analyzed the anti-nutritional factor and all the ingredients in the foods, and this is a statistical analysis, and you can actually see the differentiation between the flours, the concentrates, and the isolates. So processing does have an effect on the anti-nutritional factors. In the isolates, there are some phytic acids left, in the protein concentrates somewhat more. We also did some interesting experiments. So there's a lot of work out there on anti-nutritional factors, especially in legumes. Okay, you can quantify them. However, for one reason or another, nobody looked at it at the effect of them in vivo. Okay, so what we did, we performed a trypsin assay and exposed the trypsin to a physiological relevant concentration of the food. Okay, so diluted. And to our surprise, the effect was minor. You know, so this is basically 100% of trypsin, you know, sometimes a little bit less, less activity, but not more than 5 to 8%. So we published this, we were the first one to publish this. I'm waiting for the reaction of the scientific community. Did we make a mistake? You know, that's possible as well, but we just exposed it, we threw it out there. So my own opinion is that the effect of the anti-nutritional factors has been slightly overestimated. There's a chance there. Okay, this is my last slide on the anti-nutritional factors. What they looked at here is basically the effect of processing from the raw product to the extruded and to the cooked meal, and again the in vitro assays for anti-nutritional factors are valuable tools, really. So the next topic was led by my colleague Linda Giblin, and that's the bioavailability and the health effect of the foods. So what they chose 4 ingredients. Two of them are for the in vivo human intervention trial and some other novel proteins, and they are compared to the whey proteins, which seems to be the gold standard in terms of nutrition. We performed the in vitro digestion infogest and exposed the food to the state-of-the-art next generation gut barrier model, and that's a CACO2 HC29MTX, a co-culture. So the food, the digested food, is exposed to the layer, and then you basically look at the absorption, and we also looked at the health effect, and we concentrate on 2 effects. One is a satiety, so basically look at the expression of satiety hormones such as GLP-1, which everybody knows now. You know, 10 years ago nobody knew about that. And the muscle growth, you basically look at the growth, the muscle growth of the digested food. So here the outcomes, that's the absorbed amino acids. So 3 of the 5 ingredients, they had a very good absorption of amino acids, similar to whey protein isolate. The second one is the satiety hormones, the expression of GLP-1. So, for instance, honey chlorella, which is a yellow form of the chlorella, gives the highest GLP-1 response. And the last result is the muscle growth level. And all of them performed similar to whey protein isolate. So in general, they all performed pretty well. Now, it has been known for a while that these absorption models, they have their challenges. So my colleague Linda Giblin and Giant Leaps, together with Infogest, they embarked in a very large ring trial, 17 laboratories, because they noticed that if you performed the experiment in Italy or in Portugal, you couldn't compare it to the results in Ireland. That is, of course, that's no good, really, you know? So the challenge was to reduce the inter-lab variability, look at it, is it relevant to humans, and is it suitable for food digester. So these are the parameters of assessment: barrier integrity, transport, differentiation to inflamed barrier, and suitability for food. And if we can take this huge amount of work, just a take-home message, they were able to reduce the variability, number one. Then it takes 21 days to grow this. So after 8 days, they were able to say, go ahead, watch it, or stop. And that's a major step. And it was confirmed that it's relevant to food, so you have to deal with food. So the last couple of slides, only 3 slides left, that's on the human intervention trial. So INRAE, AgroParisTech, Claire Gaudichon, the team, they performed a human intervention trial. And as mentioned earlier, we needed labeled fava beans. And during the project, it became apparent that unless we paid somebody an insane amount of money, we were not able to produce these, okay? And I asked my colleagues in Valpro Plus, Ewan and Sheila Alves, who is somewhere here, and their response was, yes, we can, without actually knowing, could they really do it? But, you know, it's a real can-do attitude, and we really appreciate it, and we produced 15 kilograms of labeled fava beans that was necessary for the trial. That was sent to VTT. They dry fractionate into the concentrate and produced high moisture extrusion. They produced these meat analogs. The second food that we looked at is honey chlorella as a yellow chlorella form produced by Green Collab, again labeled. They used a high pressure homogenization on our recommendation, and that powder, these two foods were basically used for the human trial. And the human trial is basically Claire Gaudichon and Corentin, they're real experts, they're world expert in that area. There are only a handful of people that can do that. So they look at they put a tube through the nose, through the stomach, all the way down to the small intestine. What you could just see here, this was honey chlorella from the small intestine through the tube, you know, and that was basically analyzed then, and together with the data from the urine and from blood, you could assess the amino acid digestibility and the protein score. And here on the left-hand side, you see the amino acid digestibility of the two labeled food. That's the fava bean extradate and the honey chlorella. And you can see they're pretty high, right? The fava bean extradate has a digestibility of 84, which is just under 85, so it is classified as a moderate amino acid digestibility, but 85 is actually good, you know, 85 is good. So honey chlorella has a high digestibility. If you drill down to the individual amino acids, so there are some shortcomings, so the fava bean extradate here has less sulfuric amino acids, okay, so it falls short in being a complete protein, but I always say there's nothing wrong with the incomplete protein because we don't just live off one protein source. It has to be part of a healthy diet, and you have to mix and match. So in general, that was a really good result. So that's my last slide, and you've seen that before. The methods that we developed, we have some very good methods. We improved the protein digestibility method, and that is going forward as a ISO method at the moment, at the end of the year. Linda developed the absorption method, and another method, intestinal method that I didn't mention, was developed in Italy. We produced an enormous amount of data. So one of the deliverables was what we call a nutritional passport, which is basically a dossier of all the different ingredients of food with everything that we have. So the document is just enormous. Of course, we have the human intervention trial, and in terms of outreach, you know, we have a good outreach webinars. We have a masterclass that's published on our YouTube channel, and two members, myself and Claire Gaudichon, were invited to the FAO working group on protein quality, and there we are talking about the different methods of assessment. So overall, it's a very good result, and that's my last slide, and I thank you for your attention.
Very much. Now I will ask Mark to join the stage, and Emanuele as well, please. We are a bit short of time, but we still like to have at least one question for each of the speakers. So questions are very welcome.
Hello. Excellent presentations. Thank you very much, all of you. My question is for Mark, actually. When will we achieve price parity between fava bean isolate and whey?
Apologies, can you just repeat that?
Yes, when we will have cost parity between fava bean isolate and whey protein.
Oh, I don't want to tell Vladimir the bad news, but it took 17 years, I think, for whey to become even acceptable into, so it was a development, so it'll be a long time. No, it's an interesting one. I suppose we're looking at it from a different perspective, that we're not just taking the protein itself, and André has shown, you know, results that are coming through Giant Leaps as well. The difference in digestibility now is not as big maybe as people might think. But it's also to do with what your co-product. So in other words, your starch and fibre and so on. So for the fava bean, the business case to stack up, you need the other ingredients as well. But whey, yeah, I mean, it's proven, it's there, it's used in nutritional beverages for years and so on. So it's going to be a little bit while before it catches up, I think. Yeah, but having said that, it doesn't have to be fully functional in that point, because we recently did trials where we added fava bean into the skim milk powder. So the dairy industry are interested now in hybrids. So it doesn't have to catch up. So there could be a hybrid there between whey and fava bean, I think, which is something we're working on at the moment. Yeah. But it'll be a while, I think, before it catches up.
Thank you very much, Mark. Additional questions? If not, maybe I have one for Emanuele. So you present lots of different products, and some of them are more difficult to do, more complex to do, need a bit more of art, and some of them are easier and they can be larger scalable. So you have to choose a winner to scale up and to go for commercial applications. Which will be the one?
Well, definitely the dairy alternatives.
Okay.
Because when we are talking about meat analogs, we need to develop a texture that basically are mimicking the real meat, but not just on the plate, but also the evolution of the color, the flavor, the consistency during cooking. So definitely the dairy alternative is the easy task.
Thank you very much. Any question for our last speaker? Okay, I also have one for you. I mean, it's quite amazing the data that you produce and the ability to compare between labs. That information will be available in open access facilities for people to use, and how can you will implement that, and how can you help us to have access to it?
Our output is always publication. There will be open access in a high impact journal, absolutely. Yeah. It is in writing at the moment, so that will be out within a year.
Okay. Thank you very much. I think that everyone needs a glass of water, so we could close now and go to a break, and you can carry on to the questions after. Thank you.
So while everybody is hydrating and trying to cool down a little bit and trying to find some shade, please don't forget we have the poster session, which is starting during the tea and coffee break, and we'll run through lunchtime as well. So we'll try and reconvene, please, at 11:15 if you can. Thank you.